Hello students, can you recall the definition of genetic engineering and biotechnology which we have seen in the previous class? Yes, the field of biotechnology is used for the production of valuable products like organic acidulants, alcoholic beverages, sweeteners and gums, enzymes, and of course, transgenic plants as the produce and transgenic animals as another produce. In this module, you will be introduced to the various techniques used in food biotechnology. Coming to the specific objectives of this module, you will be able to learn after completing this module the concept of gene expression, the basic techniques in gene engineering, and to know the various techniques involved in food biotechnology. Any product, say production of protein or enzyme, starts from the very core of the cell, namely the DNA. As students of nutrition, you may recall the classes of biomolecules where you have studied the double helical structure of DNA and further the process of DNA replication, recombination and repair. Now let us see the basic techniques of gene expression. The total DNA in an organism or a cell is called as the genome. This you might have studied in your school, isn't it? And you can recall the lessons in your undergraduate courses. Now let us see what the steps are. Transcription, mRNA splicing, translation and post-translational modifications. Coming to the technique of transcription. As you know, there is an enzyme called DNA-dependent RNA polymerase. This binds to the promoter region on DNA and it moves along the DNA molecule until it encounters a termination signal. This process is called transcription. The region between the promoter and the terminator is transcribed into an mRNA. Hope you got it, isn't it? What is mRNA splicing? Let us see the process now. It's very interesting to note that the eukaryotic gene contains coding and non-coding sequences of nucleotides. The coding sequences are called exons and the non-coding sequences are called introns. Hence, the intervening sequences, namely, the introns had to be removed to have continuous coding sequences. This process is called mRNA splicing. The enzymes are used to splice the non-coding sequences. Do you know this process is very important since faulty splicing can cause diseases. Have you heard about beta thalassemia? Yes. Beta thalassemia is caused due to mutation that results in nucleotide change at an exon-intron junction. Coming to the process of translation, all of us know it is otherwise called protein synthesis. mRNA, as you know, is single-stranded and possesses genetic code which exists in the form of a triplet code. For example, you can say it as AUG or UGA. There is always an initiation codon called AUG or sometimes GUG. The termination codons are always UAA, UAG and UGA, which act as stop signals. As you see here, the ribosome basically it binds to the ribosome binding unit called RBS and it recognizes the initiation codon. Then it moves along the mRNA until it encounters a termination codon. Now the region between the initiation codon and the termination codon is translated into a protein. Basically, this process involves various components you might have studied in your undergraduate classes as amino acids, ribosomes, mRNA, tRNA, ATP and of course GTP as energy sources and a number of protein factors. The tRNA, as all of you know, it has 4 arms and the anticodon arm binds to the codon of mRNA. Now coming to the last step in gene expression, it is called post-translational modification of proteins. What happens here? Proteolytic degradation, which means a removal of a short peptide, as in the case of insulin, from pre-proinsulin to proinsulin and then to insulin. The second type of modification is called intene splicing. What are intenes? They are intervening sequences in certain proteins which had to be removed. And the other type of modification is that covalent modifications. What happens here? Phosphorylation, that is addition of phosphate group, hydroxylation, addition of hydroxyl groups, glycosylation, attachment of carbohydrate moiety to proteins, carboxylation, addition of carboxyl group, and of course methylation, addition of methyl group. Having seen what gene expression is, shall we see the basic techniques in genetic engineering? Though we use various techniques in genetic engineering, the most frequently used techniques are agarose gel electrophoresis, pulsed field gel electrophoresis, counterclamped homogeneous electric field electrophoresis, polyacrylamide gel electrophoresis, which all of you know it's called PAGE, which is very popular technique, isolation and purification of nucleic acids, nucleic acid blotting techniques, which is another popular technique, and of course autoradiography and polymerized chain reaction. Coming to the different techniques, what is agarose gel electrophoresis? As you know, electrophoresis is the movement of charged molecules in an electric field, and all of you know agarose is a polysaccharide derived from seaweeds which is used as a gel, and DNA samples are placed in the wells of the gel surface. Once the power supply is on, the DNA fragments which are negatively charged move towards the positively charged electrode. The rate of migration of DNA depends on the shape and of course the size. Smaller and linear fragments move faster than the larger ones. The gel is soaked with ethidium bromide, and the bands of DNA fluorescence when exposed to UV radiation identifies the DNA fragments. What is pulsed field gel electrophoresis? Here the large size DNA molecules can be separated. The electric field orientation is changed so that the DNA molecules can align and migrate in new directions. Coming to the technique called contour clamped homologous electrical field electrophoresis, alternate electric currents are applied to separate DNA molecules. Of course, the DNA molecules of 200 to 300 kilobase can be routinely separated. What is polyacrylamide gel electrophoresis? You know it's otherwise called PAGE. This electrophoresis, which is commonly used, is used for separation of single-stranded DNA that differs in length by just one nucleotide. It is a very useful technique for the fine separation of DNA molecules that differ from each other even by 1 to 3 nucleotides. It's quite interesting, isn't it? Coming to the isolation and purification of nucleic acids, what are the different stages of isolation? The first step is lysis of cells. This exposes the cell to the nucleic acids. For bacterial cells, lysis occurs by a combination of enzymatic and chemical treatment. For animal cells, direct treatment with detergents are used. For plant cells, physical, chemical or enzymatic methods are used. Coming to the second step called separation from other cellular components. How do you do it? You take the cellular extract, try to centrifuge it to remove the cell debris and collect the supernatant. Now, when this is treated with phenol, of course you can precipitate the proteins. This results in the formation of aqueous layer, which is very important to us, which contains the nucleic acids. Now, you apply RNAs to remove the RNA, thereby precipitate the DNA with ethanol and isolate the DNA. Fine, you got the DNA. So how do you purify this? Basically, there are two methods. In the first method, addition of detergent called cetyl trimethyl ammonium. This forms an insoluble complex. Of course, this is subjected to further centrifugation and the precipitated is collected. This is resuspended in high salt solution, thereby releasing the nucleic acids. The second method is based on the principle of binding between the DNA and silica particles in the presence of denaturing agent, namely guanidinium thiocyanate, and of course alternatively you can use a column chromatography also. Coming to another important technique, namely nucleic acid blotting technique. What is blotting? Can you just imagine? Blotting refers to the process of immobilization of sample nucleic acids on solid support. The blotted nucleic acids are used as targets in the hybridization experiments. You may wonder what are the types of blotting techniques. They are southern blotting for DNA, northern blotting for RNA, dot blotting for both DNA and RNA, and finally colony and plague hybridization. Southern blotting. What is this technique? This technique is named after the scientist Ed Southern, who developed this process in 1975. The genomic DNA is subjected to the action of restriction endonuclease. Now, the DNA fragments in the mixture is loaded onto the page to separate DNA molecules. The separated molecules are further denatured by exposure to mild alkali and then transferred to nitrocellulose paper, or you can even use a nylon membrane. This results in replication of DNA, which is annealed to the paper on heating to a temperature of around 80 degrees Celsius. When exposed to labeled cDNA probes, the probes hybridize with complementary DNA molecules. Further, on exposure to X-ray film, this reveals the DNA bands in the process called autoradiography. What is northern blotting? It is the technique used for specific identification of RNA molecules. The method is similar to southern blotting. Here, the RNA molecules are subjected to electrophoresis. It's followed by blot transfer, hybridization, and autoradiography. Coming to the technique of dot blotting. It is a modified method using southern and northern blotting technique where the nucleic acids, either DNA or RNA, are directly spotted onto the filters and not subjected to electrophoresis. This is useful in the evaluation of gene expression. What do you mean by Western blotting? This method involves the identification of proteins. Tobin et al. In 1970 developed this technique, and its working principle lies on antigen-antibody reaction, and hence it is an immunodetection technique. Here, the protein is extracted, separated in STS page, and the separated proteins are blotted onto a nitrocellulose filter paper. The proteins are detected by specific protein ligand interactions, and the bands are detected through autoradiography. Coming to the technique of colony and plague hybridization. You must be very familiar, isn't it? This method is used along with the other methods in genetic engineering. Coming to autoradiography, it's a process of localization and recording of a radio label within a solid specimen with the production of an image in a photographic emulsion. The emulsions are composed of silver halide crystals suspended in gelatin when a beta particle or an X-ray from a radio label process through the emulsions. The silver ions are converted to metallic silver atoms. This results in a visible image which can be further detected. Another important technique called polymerase chain reaction. In short, it is called PCR. It is an in vitro technique used to generate large quantities of specified DNA. Of course, it is a cell-free amplification technique. It produces, can you imagine, copies of any DNA, say even billion copies. This technique was developed by Kary Mullis, who is a Nobel laureate, and of course this technique was developed during the year 1984. The principle of PCR is that the double-stranded DNA is denatured into single strands and subjected to hybridization with the primer, and this primer template is used for DNA synthesis further. Coming to the technique of PCR, what do you require? The components are target DNA, 2 primers, 4 deoxyribonucleotides, and an important enzyme which is thermostable DNA polymerase. Coming to the technique, as I mentioned earlier, we have repeated cycles for amplification of targeted DNA, and each cycle has 3 stages. One is denaturation, the second is renaturation, and the third is synthesis. Coming to denaturation, how does it happen? The DNA gets denatured by increasing the temperature to about 95 degrees Celsius for around 1 minute. This results in separation of 2 strands. During renaturation, at around a lower temperature, 55 degrees Celsius, the primer's base pair with the complementary regions flanking targeted DNA strands. This is called annealing or renaturation. Of course, coming to the third stage, which is synthesis, it starts with the initiation of DNA synthesis at 3' hydroxyl end of each primer. The primers are extended by joining the bases complementary to DNA strands. The temperature is kept optimal for DNA polymerase. For tag DNA polymerase, the optimum temperature is around 75 degrees Celsius, and for E. coli DNA polymerase, it is around 37 degrees Celsius. Now coming to the main techniques in food biotechnology, which we will see in the subsequent lessons, let us see now what they are. Fermentation technique, recombinant DNA technology, and the widely talked tissue culture. Coming to microbial biotechnology. Microorganisms are extremely important in biotechnology. Bacteria and fungi can be grown in large scale using bioreactors, which are large vessels that typically allow close control of nutrient, oxygen levels, pH, other environmental factors. In relation to the food industry, microbes are grown in bioreactors for one of the four relations. They produce a compound that is useful either as a food additive or as a food itself. They produce enzymes that can be used to modify the properties of foods or food ingredients. They transform a food into a different type of food, or they transform waste products of the food industry, for example, cellulose, into a less environmentally harmful products, say for example, carbon dioxide. Xanthan gum is an example of a microbe-derived food ingredient, and this polysaccharide is a useful thickening agent which is used in salad dressings and other processed foods. It is produced by Xanthomonas campestris, a common bacterium. Modified starches are another group of common food ingredient. They are produced through the use of amylases and other microbial enzymes to alter the structure of corn or potato starch. Coming to the common sweeteners such as glucose and fructose, as well as various thickeners and fillers, are also obtained in this way of fermentation. Yogurt, all of you love, isn't it? It's an example of a food that is made through microbial transformation of one food, that is milk, to another food, that is a fermented product. Brewing, winemaking, and distillery industries rely on the ability of the yeast, which we say as Saccharomyces cerevisiae, to convert the carbohydrate to the product, namely ethanol. Coming to the concept of traditional microbial biotechnology. All alcoholic beverages are produced using traditional biotechnological processes. Yeasts are added to carbohydrate substrates such as sucrose. The fungi use these substrates as a source of carbon and energy and ferment them into ethanol and carbon dioxide. Without yeast, ethanol cannot be produced, and many of the flavors characteristic of each beverage depends on the fermentation process. And all of you know, yeasts are also involved in bread making. Beverage and bread making has been quoted in the Holy Bible, so you can imagine this dates back to many centuries. Cheese, yogurt, and other milk products are also made with the help of microbes. In this case, of course, the lactic acid bacteria are used as vital microorganisms. This group of bacteria produces energy through fermentation, and one of the main products of fermentation is lactic acid. This decreases the pH of milk, which causes thickening and coagulation of milk protein, creating an environment antagonistic to bacteria that are pathogenic. Coming to the concept of modern microbial biotechnology, apart from alcohol and lactic acid, many of the enzymes, amino acids and thickeners are added to food that are derived from a variety of microorganisms. In addition to xanthan gum, glutamate is also a microbial product that is commonly used as a food ingredient. All of you know it is called Ajinomoto, which is nothing but monosodium glutamate. In addition to this, it is used in many dehydrated soups and of course in many oriental foods to add brothy flavor, which is due to the presence of this amino acid. However, it is easier to collect it from the microbes than to separate glutamate from protein and from other amino acids. Have you heard about Corynebacterium glutamicum? Yes, it's a gram-positive bacterium, naturally has the ability to secrete large amounts of glutamate. This capacity is exploited by biotechnology companies, particularly in Japan and other oriental countries. So what is basically fermentation technology? This technique makes use of instruments called fermenters or bioreactors. A fermenter is a stainless steel vessel which is made up of 316 gauge to withstand the high temperature produced during fermentation process. This fermenter is motor driven from the top and is provided with aseptic valves. To withstand the metabolic heat, the fermenter is provided with a cooling coil, and to maintain the optimum temperature of microbes, cooling water is circulated. It has of course inlets for autoclaved substrate and pure inoculum. Sterile air is passed through as a source of oxygen. The microorganisms convert the substrate, or otherwise you call this as raw material, isn't it, to the product. The byproduct may end up in gaseous substances, thereby resulting in foam formation. Hence, a physical foam breaker or soybean oil can also be used to break the foam. The fermenter is provided with a flat bladed impeller and shafts, which causes continuous mixing of medium, and the factors considered as important for the fermentation process include aseptic process, oxygen transfer, mass transfer, and the maintenance of environmental conditions, namely pH, temperature, gaseous exchange, etc. When we classify fermentation process, it can be classified into three: batch fermentation, continuous fermentation, and fed batch fermentation. In batch process, the substrate is let inside the fermenter and the product is collected after the fermentation process is over. In continuous fermentation, the substrate is continuously let in and the product is continuously collected. In fed batch fermentation process, of course this follows batch culture, except that the limiting nutrient is added in between the fermentation process. Coming to the important technique, recombinant DNA technology. This, of course, we are going to see in detail in the subsequent lessons, but let me brief the technique. what happens in the production of recombinant DNA molecule? Basically, the native DNA is isolated from the cell, or you can create a cDNA by reverse transcription process using mRNA as a template, right? Okay, coming to the second step, you're going to see what are plasmids, right? Plasmid DNA is cut open with a specific restriction enzyme, and the plasmid DNA is joined with the cDNA. So this plasmid DNA forms a basis as a cloning vector. Coming to the last step, by the use of the enzyme ligase, the cDNA insert and the plasmid DNA are joined to form a recombinant DNA molecule. This rDNA is inserted into the bacterium to create a genetically engineered bacterium. So what do you want from this? You want a product, right? This can be isolated and purified by a series of techniques called downstream processing techniques. Another important technique called plant tissue culture. It depends on the principle totipotency. What is this? A tissue cell or a plant organ is regenerated into a whole plantlet. So what do you have to do this? Initially, you select an explant. The explant may be a leaf or a node, or it can be even an internode. It should be sterilized and grown in a proper culture medium, which develops into a callus. What is a callus? A callus is referred to as an undifferentiated mass of cells, which further develops into a shoot system and of course a root system by using plant growth hormones, namely auxins and cytokines. Plant cells, organs or tissues thus can be developed into plantlets. Of course, you will see the techniques of gene transfer later in a lesson. Coming to the technique of micropropagation. What is it? It produces hundreds of genetically identical plantlets. One major advantage of micropropagation is that it ensures that the propagated plants are free of viruses and other pathogens. It is useful for the crops such as potato and banana. Coming to the technique of protoplast fusion. It begins with mesophyll cells and they are treated with enzymes that break down the cell wall. This releases the protoplasm, which has an intact membrane but it has no cell wall. So do you understand what is a protoplast? A cell minus cell wall is called a protoplast, right? So this protoplast, when added with polyethylene glycol, causes fusion of protoplasts. The membranes of two colliding cells fuse and the contents of the cells are mixed. Fused protoplasts can be induced further to reform their cell walls and then plated on agar media. Now, what do you think will happen, right? Fused cells will grow into a callus, again callus. So manipulation of hormones then leads to the production of whole plantlets. The resulting plants can be backcrossed to restore valuable characteristics that were lost during the cell fusion process. To conclude, students, I think you have understood the steps in gene expression and the basic techniques used in genetic engineering process. Further, I hope you understand the concept of major techniques used in food biotechnology, namely fermentation, recombinant DNA technology, and of course, plant tissue culture. See you in the next class. Thank you.